to prepare a temporary mount of onion root tip to study mitosis. The process by which a cell divides into two daughter cells is called division. It is a continuous process involving the successive divisions including nuclear division and cytoplasmic division. The division of the nucleus, nuclear division, is called karyokinesis, while the division of cytoplasm is called cytokinesis. Cytokinesis is usually followed by karyokinesis, which is of two types: mitosis, equational division; meiosis, reductional division.
Madam, what is mitosis?
Mitosis is a kind of cell division where two daughter cells are produced which are genetically identical to each other as well as to the parent cell. It is thus the basis of continuation of unicellular as well as multicellular organisms. It occurs in somatic cells, helping in growth and repair of worn-out tissues. Let's do an experiment to prepare a temporary mount of onion root tip to study mitosis. Our aim here is to prepare a temporary mount of onion root tip to study mitosis. Materials required are onion root tips, slides, cover slips, filter paper, acetocarmine stain, dissecting needles, water, watch glass, microscope, spirit lamp, incubator, 1N-HCl, acetic acid, methanol and formalin. Procedure: growing of onion tip. Remove the old roots from onion and put it in bottle full of water. Base of the bulb should be touching water. After 5-6 days, the roots emerge. When roots are 2 to 3 cm long, cut the extreme tip, white appearance, with a pair of scissors between 8 am to 1 pm. Now fix these root tips in 1:3 of acetic acid and alcohol solution for 24 hours. Then shift the root tips to 70% alcohol for future use. Hydrolysis of root tip. Take 2 to 3 root tips in a test tube containing little dilute HCl. Heat it for 2 to 3 minutes. When the tips become transparent and fluffy, stop heating. Drain off HCl and wash the tips with water 2 to 3 times to remove the traces of HCl. Keep the roots in oven at 70 degrees Celsius temperature. Staining and mounting. Place the root tip on clean slide. Add a drop of acetocarmine. Warm the slide for 30 seconds on a spirit lamp. On warming, the stain evaporates. So put a fresh drop of stain on material. Repeat this process of alternate warming and cooling 3 to 4 times. This gives better staining. After removing the previous stain by blotting paper, put a fresh drop of stain over the material and put a cover slip over it. Keep the slide in folds of blotting paper. Very gently apply pressure with thumb over the cover slip. This gives uniform spread of root tip cells. You may crush the root tip by back of needle or scalpel before staining or mounting. Look for various stages of mitosis in the stained nucleus in high power of microscope. Identify the stage and draw diagrams. Observations. In root tips, various stages of mitosis are seen. A. Karyokinesis. Interphase. Stage in between two successive cell divisions and hence a non-dividing phase. Nucleus large and distinct with nuclear membrane. Nucleus clearly visible. Nucleus contains chromatin network. Prophase. Nucleus enlarged and occupies most of the cell volume. Chromatin network gets condensed and appear as long thread-like structure called chromosomes. Each chromosome consists of 2 or 4 chromatids held together by centromere. Nucleolus gradually disappears by the end of prophase. Nuclear membrane starts disappearing. Metaphase. Nuclear membrane completely disappears. Chromosomes become shorter and thicker and hence become distinct and clearly visible under the compound microscope. Chromosomes orient themselves towards the equator with their centromeres arranged on the equatorial line, forming metaphase plate. Out of 4 chromatids, 2 faces towards one pole and other 2 chromatids face the opposite pole. Series of spindle fibers attach the centromeres to the opposite poles. Anaphase. Centromere of each chromosome divides into 2 so that each chromatid pair gets its own centromere. Spindle fibers gradually shorten so that each chromatid with its centromere is pulled towards the respective pole, with the centromere proceeding towards poles and chromosome arms trailing behind. Each chromatid pair attached to the centromere now behaves as an independent chromosome and is thus known as daughter chromosomes. Telophase. At each pole the daughter chromosomes start uncoiling, elongate and become thin and invisible. Thus, chromosomes are again reorganized into chromatin network. Nuclear membrane reappears. Nucleolus gets reconstituted. 2 daughter nuclei are formed at the 2 poles of a cell. The 2 daughter nuclei are similar to the parent nucleus both quantitatively and qualitatively. Cytokinesis. In cytokinesis, the wall is formed across the cytoplasm and each mother cell is divided to form 2 daughter cells with diploid nuclei. Result. The cells of root tip or shoot tip are actively dividing and undergo mitotic division. Precautions. Base of onion should be in contact with water during the growth of roots. Filter the acetocarmine stain before use. Material should be warmed gently. Do not allow the solution to boil. Clean the slide and cover slip thoroughly before and after use. The slide should be neat and clean. Material should be mounted in the center of the slide. There should be no air bubbles under the cover.
Thank you, ma'am. You have given a great knowledge through this experiment.